This story that I'm going to tell, or these stories, are about Pinus cembra, species that we consider very emblematic, which occupies the timberline ecotone. This is a foundation species in that area and occurs across the European Alps and in rather disjunct places in the Carpathian Mountains, as you will see later on a distribution map. The species is obviously long-lived and quite resistant. We consider a generation time of about 40 years. And having such a long generation time in old individuals also gives us the opportunity to look into the past of the species using extant individuals, as you will see later on. One obstacle, though, to genetic work is the large genome size of 29 gigabases, which probably does not explain why I'm showing you some microsatellite data still, but I hope you excuse for that. From the biology, it's very important to be aware that the species is mainly outcrossed, and it does so by large distance wind pollination, and then the pollinated seeds the seeds coming from the pollination, they are dispersed by the European nutcracker that you see here in this picture. And I would like to show you this video that I received from a colleague in Frankfurt, which shows how the bird who has collected seed, stored them in its pouch underneath the head, and then one by one buries them in caches in the ground. And that's how Swiss stone pine is actually colonizing. And you can imagine, or as you see here in the picture, this largely happens in the main distribution range where the adult trees are, where the bird has its nest and its territories. So colonization of high elevation is to some degree hampered because of the behavior of this bird. And I will show this movie again in the end if you wish to continue watching. So this is what this German group found in two valleys near Davos in Switzerland. They did experimental work, observational work, and modeled this along an elevational gradient, which then shows that at the colonization front, the number of caches and juvenile plants is actually quite low. So it is difficult for the species to move on at in a short time period. So, because I'm recalling the past 2 decades of our research here at WSL on the species, I wouldn't want to go into much detail, so anything you want to know technically, methodologically, in more detail, you can go to the literature. But the talk that I'm presenting here covers 4 topics. Seed dispersal is very briefly already mentioned. Then I will show you data on our perspective of the post-glacial history of the species. I will show you how neutral and adaptive diversity relate to peripherality and to habitat suitability. And finally, I will show how allele frequency shifts relate to the genomic vulnerability of the species in the long term. I have to work with 2 monitors here, so it's a bit complex here. So here's the range and the sampling that I'm talking about. You see the black dots. They show you where we have samples from for the post-glacial history inference. So it's 147 populations, nearly 3,000 individuals that we genotyped here. And the colored larger circles indicate the populations that we used for the other 2 studies that I will present here. So let's start with the genetic structure and diversity part. Here you see the result of structure analysis at the presumably most reasonable number of clusters, k equals 5, and you see a nice east-west alignment, or west-east, whatever, of these 5 clusters that we observe. And you might notice that the Carpathian populations are linked to the far eastern populations of the Alps. If we go back to only 2 clusters of the same data, same analysis, we see the main split of these populations in the area between, or a bit east of Switzerland, in the westernmost part of Austria. And when we look at chloroplast markers, which we also genotype for these individuals, and we pick those clusters out of 9 that have their center in the west versus in the east, and we overlay this. Here's the break, by the way, yes. Here's overlaid on the nuclear marker pattern, the chloroplast pattern. So those clusters that have or dominate the western part of the distribution, they go all the way up from the west to this contact zone here. And likewise, those chloroplast clusters that are predominant in the eastern part of the distribution obviously also have their limits at this contact zone. So this obviously indicates that there's a clear separation of these 2 groups, east and west, a clear link between the Carpathians and the eastern part of the Alps with very low differentiation values. And interestingly, if we look at the pattern that we found in alpine herbaceous species that we looked at in the previous project, we see that the majority of the genetic breaks in these 12 species that we looked at is very close or in the same area as the break that we see in Pinus cembra. So there seems to be really a landscape-related, a topography-related, and colonization-related barrier or an area of contact which we relate to, in part at least, to the topography of the Alps and the altitudinal range that hampered fast colonization during after the last glacial maximum. Now if you look at the pollen records, and very similar, so it looks for the macrofossils that we have, that we obtained from the paleoecological group of the University of Bern. You see that the oldest findings are in this area in the central southern part of the Alps, but also there's old findings in the central and southern Carpathians, and these are very likely the areas where the species survived the last glacial maximum and colonized the Alps from there. And this relates back to this clustering into 2 groups, which cannot directly be explained by this pollen data that dates back to about 20,000 years nearly. But I presume that there is a more ancient split of these 2 groups that predates the last glacial maximum and is responsible for this clear separation into these 2 clusters. But of course, it's not just one process or one pattern that explains it all, so it's likely a combination of an ancient separation and also colonization after the last glacial maximum that involved numerous founder events, presumably. And we hope that ABC analysis will tell us more about how that has come about. And if we look at the diversity patterns, we see that it's a largely a random pattern, so we cannot associate areas of admixture with high levels of genetic diversity in these markers, in the nuclear microsatellites. So again, here it's processes that are superimposed to the post-glacial colonization history that shape these patterns of, in this case, expected heterozygosity in the microsatellite markers. So with this, I move to the next bedtime story. This is work that has been largely done by Ben Dauphin. And here we were interested in seeing how genetic diversity, abbreviated here with GD, relates to, on the one side, peripherality of populations and on habitat suitability. And just as a bit of a methodological background, we defined adaptive genetic diversity on the basis, first, of exome capture data, from where we generated a few thousand, it was about 17,000 SNPs. And from these we determined putatively adaptive SNPs based on GMSD, pi variation, FST outliers, and the environmental association analysis. And all the rest we considered as being neutral. And we wanted to specifically look at how adaptive and neutral genetic diversity relate to these two geographical and ecological proxies. The habitat suitability was measured on the basis of an ensemble species distribution model, an ecological niche model, sorry, based on all occurrences of Pinus cembra in Switzerland. And peripherality relates to the shortest distance to the range margin. So here's an example of the results that we obtained. On the top line for neutral loci, that is all loci minus all those loci that appeared in any of the four methods that I mentioned before. And in the bottom line, the adaptive loci as we defined them based on the GMSD. And you see that expected heterozygosity in the neutral loci is negatively correlated with peripherality, but the adaptive genetic diversity is not correlated to peripherality whatsoever. So this corresponds to what we actually expected. The peripherality has an effect due to fragmentation, isolation, reduced gene flow, increased genetic drift, and we would see reduced genetic diversity in peripheral populations. And the adaptive loci, we wouldn't see why they should respond in any way to the place where the population is actually located at. When it comes to habitat suitability, the plots on the right, again, on the top we see the neutral loci. They show no trend whatsoever, whereas the adaptive loci show a negative trend, that is, populations in highly suitable areas actually show a decreased adaptive genetic diversity. And actually, this was only true for those loci that were determined based on the GMSD. So we rejected the expected hypothesis for the neutral loci because we thought that populations suitable habitat, they should be very diverse. It's not that case. And we, in part, confirmed the expectation for adaptive loci under the assumption that migration is, has a lower effect than selection. With this, I come to the final part of the presentation. Here we looked at allele frequency shifts over time and related that to the genomic vulnerability of populations. And for this study, we used 7 populations from the Swiss part of the distribution rate. It was 7 sites in the 2 main genetic lineages within Switzerland, and a north-south gradient in both areas. And in each of these populations, we sampled 3 cohorts of 20 individuals each, namely a low elevation adult cohort, at the same place, low elevation young individuals, juveniles, and then a high juvenile cohort that is around the colonization front. And this should allow us to do two main comparisons, namely a temporal comparison. So how do the old trees, the old guys, relate to what the young guys experience these days? So the old trees, based on dendro counts, were on average 168 years old. The juveniles were estimated on average 15 years old. And the other comparison was a spatial analysis for the two juvenile cohorts being at low elevation versus high elevation at the colonization front. But for this presentation, to not make it even longer, I just focus on the temporal comparison. So, because we wanted to see how these plants perform and adapted to their environment, we looked at the environment they were establishing in, so during the time when they had to start their life. So this means that we took environmental data of the present period of roughly 30 years, averaged, and that was relevant for the juvenile trees, whereas for the adult trees, we referred to the environment that they experienced to their establishing phase roughly 170 years ago. So that is during the last mini-glacial period when we had a maximum of the glacial advance, the most recent glacial advance here in the Alps. And as you can see, in the left panel, where we always compare the adults versus the juveniles in the plot that describes precipitation on the y-axis and temperature on the x-axis. There's a trend in all these 7 populations for the climate to become warmer in this direction and wetter, drier, sorry. So there was a consistent trend in temperature and precipitation along the temporal axis. And I'm missing actually the lines here. They disappeared on this graph. Well, just imagine in this case, don't know why this doesn't transmit. There's a lot of movement in the old times here, and then a gradual increase of temperature in the top row here. Yeah, too bad you can't see. You can only see the scenarios towards the end of the century, which is even higher than what we expect, than what we have today, obviously. And for precipitation, the lower part, the trend is very even across time during the past roughly 200 years, and then actually it also goes a bit down in terms of climate models. So excuse me that this doesn't display correctly. Now in this case, for identifying the putatively adaptive loci, we applied again 3 different methods here. We used I should go back, LFM and Baypass, using these continuous variables, temperature and precipitation. We used, again, the same genotype environment analysis, association analysis methods with categorical variables, in this case, to differentiate between adult and juvenile. And then we also applied a sign test for consistency in the comparison between allele frequency between these 2 types of cohorts. So only those loci that consistently showed the same trend in allele frequency difference across all 7 pairs of comparisons were considered. And again, the neutral loci were those that we did not consider as being adaptive. And we then looked at the allele frequency shifts across these roughly 150 years to see what the difference in allele frequencies were in these loci. And this plot shows the neutral loci, a bit less than 9,000 loci here. So this is the empirical data. And you can see here no, you cannot, yes. The typical L shape of the frequency distribution. And what you see also here is that for every of the populations, the absolute relative shift per year was really low. So there was really a low allele frequency shift over time in those empirical data. And this was a bit, to our surprise, maybe not for the neutral loci, but when we looked at those that we considered adaptive to the temperature change and the precipitation change, we had nearly the same frequency distributions. So we were a bit worried and thought, how can we confirm or reject this empirical finding? And for this, we engaged with theoreticians that simulated these data with various selection strengths and genomic architecture parameters. And what came out was almost the same. So in fact, the empirical data were confirmed by the simulations. And still to our surprise, there was no real difference, obvious difference in the distributions between neutral versus the putatively adaptive loci. So we took this data then to evaluate the genomic vulnerability of these populations using the approach that Christian has developed, this risk of non-adaptedness that was introduced also yesterday evening. And this is the data shown for the low juvenile cohort here. First, for the temperature-associated loci. You see on the y-axis this risk of non-adaptedness. So the higher the value, the higher the genomic vulnerability, the higher the genomic offset in the long term in view of the climate in the end of this century. These bottom lines here, they mark what we saw in the empirical data of neutral and adaptive loci in terms of allele frequency shift. And you notice that in all of the populations and in both types of climate scenarios, CO2 emission scenarios, the risk of non-adaptedness is very high for all these populations. Higher, of course, for the more stringent or more extreme climate scenarios. And still vulnerable, but to a lesser degree. Also, with regard to precipitation, these populations are likely not to be able to move to the optimum that we would expect based on our analysis in the time they have available until we reach this climate based on the scenarios. So with this, I come to the conclusions. Obviously, we see a very distinct genetic structure which relates to paleoecological history. We don't know yet the details, but we're working on this. Importantly, we think that one should consider neutral and adaptive genetic diversity as two different types of diversity because they do show differential patterns, in particular here in our case in relation to peripherality and habitat suitability, but of course also in terms of any other evolutionary process, we need to keep these two types of markers apart. We saw a surprisingly low rate of historical allele frequency shifts over several generations. So these 170 years means roughly 4 generations in Pinus cembra timing. And this was consistent in not only in the empirical, but also in the simulated data, which helps us believe in this. And from this, we derive that Pinus cembra shows a high genomic vulnerability under the predicted climate change. And we anticipate that the allele frequency shifts are insufficient to actually cope with the expected change in temperature and precipitation. So what are the take-home messages that I would like to show you here? Given that these changes in allele frequencies over time are too slow, in relation to the environmental change that we are experiencing and will be experiencing in the near future. We believe that migration is required to tackle the ecological niche, but we wonder if with this bird behavior, whether this colonization is actually fast enough to match the speed of environmental change. And therefore, we guess that assisted gene flow that is within the range or assisted migration, meaning going beyond the current range, could be required. And with our study, we hope we could be able to inform the requirements in terms of predictive genomics. So what is required? How far do we potentially need to move the species up the hill to avoid competition from low elevation species immigrating into the territory? And my take-home message actually is studying Pinus cembra in a wonderful alpine environment and with cool colleagues is a wonderful thing to do. And with this, I would acknowledge all these colleagues. Many people helped doing field sampling, also beyond the ones that I show here. We have many people sending samples, people helping in the lab, in the computer cluster behind simulation tools and so forth. And of course, financial support by the Swiss National Science Foundation, who, by the way, are also supporting our conference here, should be mentioned also.